I started this unit, this chapter on meiosis with a promise, and that promise was that we were going to understand the molecular basis for Mendel's laws. So let's make good on that promise here. So the first law was Mendel's law of segregation, this idea that homologous alleles are separated out. So now let's take a look at how that plays out in meiosis. So if we had the genome big A, little a, we would say that, okay, well, let's just say cross big A, little a. We said, all right, well, let's put one parent on top, one parent on the side, right? So that's our cross. And we said that the big A and the little a separate out, right? Big A goes here, little a goes here. Same on this side, big A here, little a there. And then we filled out the Punnett square, and away we went. But the question is, what's happening at the cellular and molecular level to explain that? Well, we can understand that. Let's bring in some candy bars. Let's imagine we are at metaphase 1 of meiosis, where homologous chromosomes are paired up. And let's suppose that the darker color Milky Way is big A, the yellowish Milky Way is little a. Well, in meiosis 1, what happens is the homologous chromosomes are separated out to opposite ends of the cell, which means half the gametes are going to get big A, half the gametes are going to get little a, which is exactly why we do this when we make Punnett squares. So we see that Mendel's law of segregation, what's actually happening is that those homologous alleles are separating out during anaphase 1. So we can see the molecular basis for where that's happening. Mendel's second law was the law of independent assortment. And we can explain this one as well, so let's do that. So let's just look at this top level of this giant motherlode Punnett square. Let's suppose we had big A, little a, big B, little b, and then the other parent doesn't matter. We'll just ignore the other parent. So what we said is that every gamete gets a big A and an A and a B, right? So we said, all right, so big A, big B. Big A could also be paired with little b. Little a could be with big B. And little a could be with little b. And so we have these sort of 4 possible combinations of gametes based on these A's and B's. So how do we account for that? Well, again, our chromosome models come to the rescue. It turns out that, again, in meiosis 1, when the homologous chromosomes line up in pairs, it's random. And so in this instance, if they're lined up this way, we have big A on the left and big B on the left. And so when they separate out, at the end of meiosis 1, we're going to have half our gametes are going to be big A, big B. The other half are going to be little a, little b. However, that's not the only way that they could line up. What if in meiosis 1, they line up like this? Equally possible, because again, the cells don't know whether we're using a capital letter or a lowercase letter, right? And so if they line up this way, then again, during meiosis 1, when they separate out, now big A is with little b, and little a is with big B. And so in meiosis 2, when these sister chromatids separate out, then that's what we're going to see, is those combinations. So we can draw that out, and I think it's worth doing. So let's just kind of draw. I'm trying to get my marker uncapped here. So I'm going to make 2 blues like that, and 2 reds like that. And then I'm going to use my pencil. Uh-oh, I'm running out of battery. And I'm going to use my pencil. I'm going to call this big A. What am I doing? Big A, little a, big B, little b. So if they line up like that, then eventually, if you follow this through the steps of meiosis, you're going to get a big A, big B, and then another copy of big A, big B. and then you're going to get little a, little b, little a, little b. However, they might also line up a different way. They may line up like this. And so now your big A, little a, is like that, but now the big B is over here, and this is the little b. And so if that's the case, now you're going to get this kind of a setup, where the big A and little b are going to be in one. And then the little a, big B are in the other. And so what we see is 1/4 of our gametes are this big A, big B. 1/4 of our gametes are little a, little b. 1/4 are big A, little b. And 1/4 are little a, big B. And so again, remember, it's not that meiosis is happening one time, it's happening millions and millions and millions of times. And so roughly half the time at metaphase 1 they line up like this, and the other half they line up like that. Here's a great diagram from your textbook that summarizes these two laws of segregation and independent assortment in the context of meiosis. And so I really think it's worth taking some time to study that and understand how these two laws are manifested through the movement of chromosomes during meiosis. The last aspect of Mendel's laws that meiosis can help us explain is sources of genetic variation. So there's incredible variation in genetics. You know, Michael Jordan and Danny DeVito are members of the same species. Huge amounts of variation in the population. And so where does that come from? How does that come about? Well, we can now understand where it comes about. In one place where it comes is from independent assortment. So each parent only passes on one homologous partner, and it's random which one that's going to be. And so this is why it's possible for two parents to have very, very different children, because they're passing on different genes to each one, and so then that leads to more variation. Another source is crossing over. So remember, there's a diagram we drew here where we kind of talked about how each individual, when they make their chromosomes through crossing over to pass on to their offspring, they sort of blend the chromosome from their mom and their chromosome from their dad into unique chromosomes that have never existed before. And so through crossing over, we are creating new combinations of genes that have never existed before. And again, crossing over doesn't really show on, in these pictures that we see, but it's absolutely there. So again, like really all of these chromosomes should be this blended mix of red and blue. But again, that's just too complicated to show on these figures, so they don't show you that. But crossing over creates tons of genetic variation. And when does that happen? Prophase 1. Okay. Finally, random fertilization. So we've created tons of variable gametes as a result of these two different processes. And so then it's just any one of these highly diverse gametes may actually end up being the one involved in fertilization. And so, some estimates here. There are about 8.4 million combinations of chromosomes based on independent assortment alone. So, one individual can make 8.4 million combinations of chromosomes based on that. The chance of forming an offspring with any particular combination of chromosomes is going to be this from the mom and times this from the dad. That's 1 in 70 trillion. And that doesn't even account for crossing over. So if you ever heard the phrase, you're 1 in a million, yeah, you're 1 in 70 trillion, which is pretty incredible. So we can even understand the huge amount of variation we see using these laws of meiosis. That's all for now. Thanks.